Okay, now this is for week 1 lecture, and we would like to go through the learning outcomes of the lecture. And in this case, we would like to understand what is quantitative genetics all about. And we have 4 learning outcomes. The 4 learning outcomes is you are able to explain basic genetic principles for quantitative genetics. Second, describe the quantitative genetics principle in natural and selective environment. And therefore, in the third learning outcome, you should be able to apply statistical techniques in quantitative genetic studies. And finally, the fourth one, apply the basic understanding and application of quantitative genetics in plant and animal studies. Now, when you are going to start on quantitative genetics, you have to have 5 assumptions that you need to remember. The first one is looking at the reference population. Most of the time, you will need to define your reference population. You need to quantify the allelic and genotype frequencies in respect to the reference population. Number 2 is you have to make sure there's absence of linkage, and whatever phenotype that you observe is actually not affected by autosomal linkage genes. And number 3 is the species of study, or whether it's animal, plant, they should follow the Mendelian inheritance, where they are diploid and the genes segregate and assort independently. Number 4, during the formation of lines or formation of the reference population, there must be absence of selection during that, and for that we can estimate the genetic variance to reference to the reference population, where there's no selection occur when inbred lines are crossed. The fifth part is we say there is no breeding of the reference population. In this case, inbreeding coefficient is 0. The analysis becomes more complex when the breeding is coupled with 2 or more loci, and if there's epistatic effect. Now, again, refresh. What is quantitative traits? It's a branch of genetics where individuals' genotypes are unidentified, traits of individual measured, genotype values are assigned to the genotypes in the population, and then the main emphasis in quantitative genetics is to look at selection in control population of known ancestry. Recording. What are you doing? Now let me give you an example of traits that is affected in terms of quantitative genetics. This is a example of Siamese cats. I think a lot of you all love cats. Now if you see okay, now let's look at an example of Siamese cats. Is one of the examples in quantitative genetics, where Siamese cat, if you look at this picture here, all right, what do you see? You see pigmentation, and the pigmentation is localized in the area of the face and also at the legs. And apparently this cat has a mutation in the C gene, and the C gene allows dark pigmentation to be formed. But however, so the CH allele of the gene is heat sensitive. So if you could see that at extreme temperature, at low extreme temperature, it becomes darker, but not in high temperature. And this is a real example of how an effect of quantitative genetics, where the expression of the gene is affected by the genotype and also environment. Now, in quantitative genetics, the traits that you will observe, normally we will do a normal distribution curve. In this case, you will see 2 types of traits. One is called a discontinuous trait, traits that have very few distant phenotypes. And the second one is continuous trait, the traits that actually have apparent continuous distribution of phenotype. If you look at height, weight, and and, uh, and other traits that you can recognize as a quantitative trait, is when you plot them, you plot the individual. Say this is a height, you will see a normal distribution where it looks no distinct categories, tends to be quantitative, controlled by a lot of genes. Okay, this is an excellent model to show that this is controlled by an environment. Now for discontinuous traits, the traits now shows here, it has a distinct characters, tend to be qualitative, is controlled by a less number of genes, and is unaffected by the environment, such as blood group. Okay, A, B, O, and right. So refresh again is a study of inheritance of continuous trait. It is called metric traits. And why some traits have continuous phenotypes? These are the reasons that make them to have continuous trait. The first reason is it has a numerous gene effect that affects the expression of that trait. The number of genotypes and thus the number of phenotypes are large and appear continuously. Whereas the second factor is the environment factor that affects the expression of the trait, and this actually produces a large range of phenotypes where we know it as a norm of the reaction. Now there's a difference between Mendelian versus quantitative genetics. It's a very simple way to look at it. One is the size of the phenotypic difference between the genotypes. Two is the individual difference within the genotype. If one is more than two, it's Mendelian. If two is more than one, it's quantitative. Right? Now just very simple mind map. There are many genes. It affects the trait, and there's a factor of environment. That is the easiest way to remember what is quantitative genetics. So lecture 1 was actually to emphasize the basic principle of quantitative genetics, identify the difference between qualitative and quantitative genetics, understand what is phenotypic and genotypic value, and of course the next step is to understand what is population mean, average effect, breeding value, dominance deviation, and interaction deviation. So as again to refresh, it's a study of control by many genes, and it's called polygenic inheritance. Okay, in plants you can look at the diameter of the stem, the leaf, and the traits have a continuous variation. And the qualitative genetics is more like our traits following the classical Mendelian, and is one gene controlling one trait, and therefore an example is like antigens and antibody, and you see colors in chicken, white, and so on. So again, if you want to tabulate and understand the difference between qualitative and quantitative genetics, qualitative genetics, it deals with inheritance such as color types and forms, whereas quantitative genetics is more to height, weight, and length. And qualitative genetics is discontinuous type of variation, whereas quantitative genetics is displayed by normal distribution. Each qualitative trait is controlled by alleles of a single gene locus, and each trait is governed by many allelic gene loci, and the phenotypic expression of trait is not influenced by environment. But here in this case for quantitative genetics, environment conditions affect the quantitative traits variously. In qualitative genetics, it concerns with individual mating and their progeny. In quantitative genetics, is more to many matings of individuals in a population. Analysis of data is made by counts, and analysis of data in quantitative genetics is made by quantitative methods. Okay. Alright, so to summarize, variation without natural discontinuous is called continuous variation. Characters that exhibit discontinuous variation is called quantitative characters. This branch in studies that metric characters outline is called quantitative genetics. So just for a refresh, what is a quantitative character? What is it? So you will say a quantitative character is a trait that shows continuous variation and is controlled by many genes. So therefore, when it's controlled by many genes, it's called polygenic inheritance, and it is a value that is controlled by genotype and environment, right? Okay. Number two, list three difference between qualitative and quantitative genetics. I'm sure you saw the table, all right? Qualitative controlled by single gene, quantitative controlled by many genes, all right? Second, qualitative is discrete, it's discontinuous, quantitative is continuous. Number three, qualitative is uh, analyzed through qualitative measures where based on phenotypes, all right, whereas quantitative is measured by quantitative methods where you need to have statistics, all right. There's another three more, okay. And in that case, a very simple refresh question, if you're looking at genes, you need to understand the concept of dominance, codominance, and overdominance we covered in population genetics. So we will be discussing more of this in the next lecture. So do read up what is dominance, codominance, and overdominance. So we're now going to take a short quiz on understanding what is continuous variation for the first week of lecture. All right. Have a nice day.